Hello students, in this session we're going to discuss about the transgenic approaches in plants resistant to viral infections. You already know that naturally plants have different defense mechanism to resist the viral infection. Whenever a plant got wound, so they will secrete different types of chemicals like you can observe salicylic acid, jasminic acid, nitric oxide, and even secondary metabolites, polyphenols will be secreted from the plants, so that can easily defend the pathogens of the viral infections. But in some occasions the virus has the uphand as compared to the plant defense mechanism that will infest the plant successfully and then break out the barrier and infect the plants and cause diseases. And if it is an agriculture plant, it will reduce the yield, it will reduce the photosynthetic activity, and so on. Because of that, many scientists goes with a transgenic approach to enhance the defense mechanism in plants against virus infections. So these are the approaches we need to focus on in transgenic plants. If some plant is susceptible for some any virus infections, we need to focus on these parameters, and then we need to choose one of that effectively that neutralize the virus infection. So in that, these are the several approaches. If you take the first one class, protection mediated and pathogen derived resistant, and then it contains other type of things which mainly present in the pathogen. So whatever the pathogen that contains, that will be focused, and then we need to prepare a defense mechanism against that, so that effectively neutralize the infestation of that virus. And even we can also focus on different other mechanism that targets the DNA, mRNA, and then we can also use precise genome editing technology to counteract the virus infection. So we'll go one by one, the whatever the parameters here, the approaches with respect to virus infection. So we need to go for a transgenic approach to get rid of virus in the transgenic plants here. The first point is cross protection mediated resistant in transgenic plants. This is the simplest approach. We can use this in different plants. I can observe that a virus which has virulent gene and causes pathogens in plants, we will design our own virus in such a way that it has, virus generally has two traits. One is called as the antigenicity, and second one is called as pathogenicity, right? So pathogenicity, whenever we neutralize the pathogenicity and retain antigenicity, these whatever the modified virus become milder, which will cause mild infection, and then plant can defend easily in secondary infection. Whenever the true virus comes out, it's like a defense mechanism in animals, like a attenuated vaccine. Same way, you can observe that we can prepare a milder type of virus that can cause some immunogenic response in plant, but not cause any harmful effect. And further, what happens? Actual pathogen enters. A plant already has a defense mechanism that will trigger out and then successfully defend the virus pathogen. You can see here, this plant is infected with a mild or attenuated virus. Because of that, this plant in further infection, these are the vectors like aphids will take up the virus and then cause infection. So because of that, whenever aphids carries the virus infects to these the plant treated with attenuated mild type of virus, definitely plant trigger out the defense mechanism, and then the pathogenicity of the virus will be neutralized. But you can take that normal plant which is infected, definitely that will not produce yield, and then that will be affected. This is the mechanism. We can use it for animals as well as plants here. So we can use a milder type of viruses that make them to infect the plants, and then the plant will have a defense mechanism, and then further plant truly defend the actual pathogen when enters in secondary tract. This is the mechanism that can use for different types of creeper plants. Next type of pathogen-derived resistant is called code protein genes. The virus gene will be expressed and integrates into the host genome, and then further it is zombified the genome of the bacteria, and then it will express the code protein as well as the genetic material. Right here, we'll focus on we'll target on the code protein in such a way that we'll insert a code protein, antisense RNA of code protein in the plant genome. Whenever the actual pathogen enters, what happens? It will express and produces, integrates into plant genome. And whenever expression starts, here it will produce a transcription gene, mRNA, for the genetic material that will express and produce genetic material. But it need a code protein to rupture and then come back from the host plant, right? So we already introduced a antisense RNA of the code protein. Whenever that gene expresses, mRNA is expresses, the antisense mRNA also expresses, and then these two sense and antisense will complement and forms double-stranded mRNA. That double-stranded mRNA will be spliced by DNases in the cytoplasm. So that's why that in this way we can neutralize the virus infection by targeting the antisense code genes that is integrates into host genome. And one more target protein that is there in the pathogen is called movement protein. This movement protein is a protein that is produced from the host, that is produced from the pathogen, that helps in the movement of this pathogen through plasmodesmata connection. This movement protein with RNA will move from host to host through plasmodesmata connection. Whenever we produce, we'll target a movement protein and produce, will produce an antisense RNA for this movement protein. What happens? It will express and produce movement protein mRNA, and then will again the cell produces antisense movement protein, and then they will complement to each other, and then they form double-stranded mRNA, and then that will be neutralized by the nuclease activity further. So like that we can target the antisense RNA of movement protein, and we can neutralize, and then we can prevent the movement of these pathogen into next neighboring cell through plasmodesmata connection. Next way we can introduce a transgenic plant, that we can introduce a plantibody with respect to the virus. Virus will have epitope, right? You can see here virus will have a code protein, and they will have a spike on the surface of that, right? This spike protein act like a epitope. Epitope is a specific region in on the antigen possessing pathogen. That means pathogen possesses this antigen. This antigen contains a specific binding site called as epitope, right? Against this epitope, the antibody in the animal as well as plant produce. So this generally antibodies are not produced in plants, but we can trigger out a defense mechanism, so that it will produce a protein that resembles the antibody here. Antibody will have a specific protein, a specific site of region, that is called paratope, which complementarily binds to the epitope of the pathogen. Right, we'll express a specific anti-immunoglobulin in plants, that's called plantibody. Plant produces antibody that binds to the epitope of the pathogen virus, is called as plantibodies. Whenever the plantibodies are produced that complement to the epitope of the pathogen, and then that will be neutralized further. Likewise, we can neutralize the pathogen, and definitely this is a way we can neutralize the viruses in the infection by transgenic methods. The fourth method is called as cDNA of satellite RNA. You can observe that in some viruses, some satellite RNA is expressed. Satellite RNA is expressed. So what we will do is we'll complementarily produce the mRNA for this RNA. What happens? Whenever this satellite RNA is expressed, will complementarily will produce antisense mRNA here. This is called cDNA, so that it will be neutralized by nucleases further. Like that, we can target the satellite RNA also in degradation, and then reduce the infection of the pathogen here, virus pathogens here. And next mechanism is called as defective viral genome. What happens in this process in transgenic approach means we'll insert a modified genome that resemble the viral genome, but that is defected because of the mutation, because of some other type of aberrations. Whenever you introduce a gene or gene that belongs to viruses into the plant genome, what happens? Actual pathogen enters, and this genome will be integrated, right? Whenever that integration happens, these expression of these defective viral genome also happens, as well as this expression of the actual true pathogenic genome also happens. So because of this, there is a competition for the active site, for the expression. So because of that, we can easily combat the infection of these viruses. We can target by defecting, by mutating the viral genome and insert into the host genome, and then they become transgenic and then produce effectively a defective protein or expression, and that will neutralize the actual virus genome expression. And next way of resistant in transgenic plant is we will target a ribosome mediated production. What is ribosome? Not ribosome, it is ribosome. That is a specific enzymes which mainly target ribonucleic acid. Ribozyme means, in the name itself says that zyme means enzyme, and then ribo means it will mainly focus and target the RNA. It's like RNases. Whenever a virus RNA enters, what happens will trigger out these ribosome in such a way that this ribosome block the translation of these virus genome, and then this oligonucleotide of the virus will not be expressed, and then virus, these ribosomes specifically target the RNA of the virus, and then it will cleave out and prevent the replication and translation of the viruses further. So this is the way we can target and nullify the action of the virus infection here. And one more effective and approachable type of resistant, transgenic approach to get resistant to viral disease is antisense RNA technology. This is the common procedure which can be do. We can target double-stranded RNA virus or single-stranded RNA virus, or double-stranded DNA virus in both ways. What happens in this process? It's called antisense RNA technology, or gene silencing, or RNA interference technology. Whatever they ask, you can write this one. Whenever double-stranded RNA and double-stranded DNA is there, we can observe that some risk complex will recognize these foreign DNA or RNA or mRNA, whatever. Whenever risk complex recognize that, it will assemble some proteins, right? The proteins called dicer proteins. Whenever dicer protein recognizes, it will attract the risk complex. This risk complex mainly splice the single strand RNA, double strand RNA, and then what happens? Definitely, that will degrade the target mRNA and then silence it, right? And antisense RNA technology means whenever you can see this is the sense strand of virus enters, what happens? We'll integrate the antisense of RNA into the plant genome, right? So whatever virus will express and produce a sense strand, and then plant genome also, because of the integration, it produces the antisense and then produces a complementary mRNA, and it becomes double-stranded mRNA. Whenever double-stranded mRNA is recognized, these dicer complex will recognize and attract the risk complex. This risk complex binds to the and non-labeled. You can observe that the native mRNA will have capping and tiling, right? The 7-methyl guanosine cap, and then poly A tie will be there, right? Poly A tie will be there. This act like identity code for recognition. So this recognition will not be there for this double-stranded mRNA that is complementary to the virus, and then they will be degraded by risk complex, and then the expression of those viral RNA will be silenced. And one more method is called replicase mediated. Replicase mediated means this, it's an enzyme that produced that helps in the replication of the host pathogenic virus that contains the genome of the pathogen contains these replicase that helps in the replication of the virus DNA. When you target these replicase enzyme, what happens? The replication of this virus genome will not happens in the host organism. So this way we can neutralize the virus pathogenic pathogenicity in the host plant. And one more effective method up to now is called as CRISPR-Cas9 or TALEN method of precise genome editing technology. What happens whenever double-stranded RNA virus or single-stranded RNA virus enters? What happens? You know, will design a CRISPR-Cas9 technology in such a way that the pre-cRNA inside that is complement to the pathogen, whatever the pathogenic virus enters, that is complementary to the viral DNA. So whenever this effector complex of the CRISPR-Cas9 will recognize the virus DNA, what happens? That will splice out the viral DNA in a small pieces, and then stop the integration of this viral DNA into the nuclear genome of the host plant. Same with RNA virus. RNA enters will design the pre-cRNA and then the and this tracer RNA, and they go together called as a small guide RNA. A small guide RNA will design, and then that should be complement to the virus particle. Whenever that happens, definitely RNA enters that start replication. This Cas9 nucleases will bind and then degrade the RNA of the virus particle. That's the way we can neutralize the integration of the virus genome into the plant cell. Next methodology is called as RNA mediated type of viral resistant. This is the second method with respect to homology dependent resistant. So if you observe that whenever RNA enters, what happens? Different proteins will recognize this, and then will stop the transcription. Before the transcription only, whatever double-strand DNA or single-strand DNA or mRNA of the virus will be neutralized because of this the protein which is associated in formation of risk complex. So dicer will be recognized, and then along with that, some other proteins will recognize that, and helps in stopping of this expression. That is called post-transcriptional gene silencing. Before the transcription only, the viral gene is silenced. So this is the way we can easily neutralize the virus integration into plant genome. And one more mechanism is transcriptional gene silencing. So post transcription means after transcription, whatever the mRNA, matured mRNAs will be there, they will be targeted and then they will be neutralized. And second method is called transcriptional gene silencing. Is there, in transcriptional process only, we can neutralize by the formation of risk complex, and then the protein and other associated proteins will be binds to the viral genome and then neutralize by the process of endo and exon nucleolytic activity. So that's the way we can neutralize it.